So thank you, Tran, for the introduction. So good morning, everybody. So this is Aiping from Berry Genomics, and today I'm going to talk about promising clinical application of long-read sequencing for human genetic diseases. So founded in 2010, Berry Genomics is dedicated to large-scale clinical applications of high-throughput technologies by providing one-station solution for genetic screening and diagnosis of human diseases. So from 2011 to 2017, Berry Genomics has commercialized short-read sequencing-based assays like NIPT, CMV-seq, and WES for prenatal, perinatal, genetic screening, and diagnosis. So since 2017, Berry Genomics has really focused on the clinical application of long-read sequencing. So we have tried Sequel, Sequel2, Revio, and now Vega benchtop system. So Berry Genomics was among one of the pioneering companies that commercialized NIPT since 2011. So we have provided one-station comprehensive solution for NIPT, which enables the laboratories to run the assays locally. So far, our NIPT assay has been adopted by over 500 prenatal centers in China. So over 8 million tests have been performed. As a leading company in clinical genetics in China, Berry Genomics has really focused on the clinical demand for carrier screening, prenatal screening, newborn screening, genetic diagnosis, and the PGT area. So we are pioneering at the large-scale clinical applications of short-read sequencing assays and now long-read sequencing. So we are technology-driven and highly value the quality of all the assays we have developed. So the initial reason that we need to find alternative technology other than short-read sequencing is because of the existence of challenging single-gene disorders that are difficult for short-read sequencing. So there are about 400 genetic disorders that are very complicated structure-wise or have very complicated variations. So among those, the top famous ones are, thalassemia, congenital adrenal hyperplasia, SMA, and Fragile X because of disease severity and high incidence. So with the determination to comprehensively characterize those genes, Berry Genomics has sought to adopt HiFi long-read sequencing since 2017, and then start a long-time journey of collaboration with PacBio. So we highly value the strategic partnership between PacBio and Berry Genomics. And me personally, I have been really enjoying working together with PacBio teams. So early in 2017, we started to explore clinical application of thalassemia in the Sequel platform, which led to the publication of a first long-read sequencing-based thalassemia paper worldwide in 2020. And with the success of those, we started to invest heavily on the Sequel2 platform in 2019 to 20. So in 2021, in collaboration with Dr. Deshen Liang, we launched the Thalassemia Gene Atlas project and led to a series of publications on the carrier screening and genetic diagnosis of thalassemia, which led to the final commercialize of thalassemia assays in 2021. So by 2023, we have done over 200,000 clinical tests for thalassemia assays. And we also publish and commercialize RNA-based assays for SMA, CAH, Fragile X, hemophilia A, ADPKT, etc. We also start to adopt Revio system for both technology service and clinical application of long-read whole genome sequencing. So through the early access agreement with PacBio, we get access to two Vega systems last year, and we start to explore and transfer all the target assays in the Vega system. So this year we get 3 more Vega units and 5 on the way later this year. So we have finished transferring all those assays to Vega system. So methodology-wise, we use multiplex long-range PCR to get the target for single gene disorders. We use target capture for the panels, and we use the conventional mechanical sharing for WGS. So these are suitable for all different kinds of samples. For single genes and panels, they work both on SQL2 and Vega systems. And for the WGS, we can use either Vega or Revio system. So these are the data comparison between Vega and the SQL2 systems. So there are 2 sets of head-to-head comparison, which means the same libraries were sequenced in parallel in 2 systems. And then there are multi-batch comparisons. So from the data comparison, we can see that Vega has shorter running time, generate more 2 to 3-fold of HiFi reads, and have higher sequencing quality. Vega also excels at data processing efficiency and less hands-on time. So over 300 tests have been performed from the head-to-head comparison, and both Vega and SQL2 get identical and correct genotypes. And now after intensive over 3,000 tests for over 10 assays, including thalassemia, Fragile X, SMA, CAH, etc., on 12 Vega cells. I think now it's safe to conclude that Vega is a perfect platform for clinical applications of target assays. We also have some head-to-head comparison of Vega versus Revio for WGS data generation. So this one is for HG001, which is the standard cell line. So Vega generated about half HiFi reads of Revio system. So it's 65 gigabytes. With a similar quality comparable to Revio. So for one cell, we can get a little bit more than 20 times depth of whole genome sequencing, which is very decent for clinical whole genome sequencing. Also, for the methylation, we can get a very similar result using the same amount of data as Revio. So as represented here by the imprinted control region correlated to Prader-Willi syndrome and Angelman syndrome. So our preliminary data has suggested that Vega could be a promising platform for clinical whole genome sequencing. So for the clinical, for the large scale clinical, so far we have get over 400,000 tests performed by HiFi long read sequencing technology, and the number is just keep increasing. So as I mentioned earlier, Berry always want to provide a one-station solution from sample to report for our customers, which include sample preparation, the reagent kit, the instruments in collaboration with PacBio, and the analysis software, automatic reporting system, and the genetic counseling team. So which is empowered by TGS Pioneer, which is the bioinformatic pipeline for accurate variant analysis, and GenomeEasy, which is our recently developed AI-assisted system for efficient data annotation and interpretation. So lastly, I want to make a short introduction to Axolome, which is a subsidiary of Berry Genomics located in Hong Kong. So Axolome is a leading company, a leading laboratory for clinical genetics, which was co-founded by Dr. Dennis Lowe, who discovered cell-free fetal DNA and is the inventor of NIPT. So Axolome runs the international business of Berry Genomics, and Dr. Kenneth Kam is the international market director. So can you stand up, please, Dr. Kam? So thank you. So Dr. Kam will have a talk in the breakout session tomorrow afternoon. So you are welcome to join the session. So I'll stop my talk here. Thank you, everybody.